welcome to the deep dive. Today, we're looking at something really quite fundamental, a potential breakthrough in how we detect and analyze single molecules. Think DNA, proteins, even things like viruses.
Right. And for years, the big goal, the mission, if you like, has been getting both incredible speed and really fine detail at the same time.
Which sounds simple, but it turns out it's incredibly hard.
It really is. And it's always run up against this, this hard barrier for the last, 2 decades? Especially with solid-state nanopores, people like them because they're stable, chemically, mechanically.
Robust.
Exactly. The whole field's been built on one main technique, ionic current blockade-based sensing.
Okay, unpack that a bit. Ionic current blockade.
It's elegant, conceptually simple. You have this tiny pore, you apply a voltage across it, ions flow through.
Like a tiny current.
Precisely. Then your target molecule, DNA, whatever goes through the pore, it blocks some of those ions temporarily.
So it disrupts the flow.
And that disruption, that dip in the current, that's your signal. That's how you know something went through.
And that basic idea, that's what powered the first generation of, say, nanopore sequencing devices.
Absolutely. It drove commercial technology. But, and this is the key point, if it was so successful, why are researchers now basically saying we need to rip this out and start again?
Yeah, why throw out the strategy that got us this far?
Because that elegance, that simplicity, well, it's kind of run out of road. We're diving into this really interesting 2025 research article today. It argues that the ionic current method has these fundamental physical limits we just can't engineer our way past.
Okay.
And the proposed solution is a totally different approach, a real paradigm shift. It's called Nanopore Electrometry.
Nanopore Electrometry. Sounds different.
It is. So our mission today is to figure out, why did the old method hit a wall, especially with speed? How does this new electrometry thing work? And crucially, what new possibilities does it actually unlock?
All right, let's start with the problems. The failure of the past, as you put it. If ionic current blockade is still used commercially, what are these limitations, these fundamental roadblocks?
Well, the paper identifies 3 core issues, and they're really baked into the physics of measuring that bulk ion flow. The first big one is this painful speed resolution trade-off.
Okay, speed versus resolution. Why can't you have both? Why can't we just measure faster and still get a clean, detailed signal?
It's about the signal versus the noise. Imagine like trying to measure water flow through a pipe. If you want a really accurate reading, high resolution, what we call a good signal-to-noise ratio, SNR, you need to measure over a certain amount of time. Let the signal build up.
Okay, average it out a bit.
Kind of, but the faster you try to sample, the shorter that measurement window becomes.
Yeah.
So fewer ions, fewer charge carriers actually contribute to each individual measurement point.
So the signal itself gets weaker relative to the background noise.
Exactly. It just gets drowned out. The study really confirms the practical limit. Where you still get a reasonably clean signal with this method, it tops out somewhere in the range of only a few kilohertz, a few thousand measurements per second.
A few thousand per second? That sounds fast in human terms, but maybe not when you're talking about molecules zipping through a tiny hole.
Precisely. And that brings us straight to the second major problem, this huge speed disparity.
Okay.
When you use an electric field to pull a molecule, like DNA, through one of these solid-state nanopores, the speeds can be, frankly, ridiculous. We're talking translocation rates potentially up to 1 million bases per second.
Whoa, hang on. 1 million bases per second. Let's put that in perspective for you listening. That's like reading a decent-sized novel, maybe 300 pages in less than 1 second.
Yeah, it's blisteringly fast. And that speed, 1 million bases a second, just completely dwarfs the few kilohertz measurement limit of the ionic current method.
So the molecule has already flown through the pore before the detector could even get a good look at it.
Pretty much. Some techniques use biological pores, things with motor proteins, to deliberately slow the DNA down, maybe to 500 bases per second. That helps.
Right, controlling the speed.
But if you want really fast detection, using these robust solid-state pores, that speed mismatch is killer. It means most of the identifying features of the molecule, the sequence information, the structural details, they just fly by completely undetected.
You might know something went through, but you have no idea what it really was in detail.
Exactly. You get a blip, but not much information.
And if you can't slow the molecule down enough, and you can't speed the measurement up enough, That lack of resolution must really limit the kind of signals you can actually capture.
It absolutely does. And that's the third big limitation, limited signal complexity.
Meaning?
The signal you get from ionic current blockade,
Hmm.
it's mostly determined by just the physical volume the molecule takes up in the pore, how much space it blocks.
So bigger molecule, bigger dip in current?
Roughly, yes. It correlates mostly to the average volume or the average charge density along the molecule, but it lacks the sensitivity to pick up on, say, subtle differences in how charge is distributed along a protein chain or specific bumps and shapes.
Which might be okay for something relatively uniform like DNA, maybe. But if you're trying to analyze more complex things,
That's exactly the issue. For the really cutting-edge stuff like protein sequencing or glycomics analyzing complex sugar structures, the molecules are much more varied. Complex 3D shapes, patches of positive and negative charge all over the place.
right.
The standard ionic current method just doesn't give you enough detail. It gives you like a blurry, averaged out picture. It struggles to distinguish those finer points.
Okay, so it sounds like the old method, despite its initial success, has really hit a fundamental wall. Speed and detail just don't mix well with it. So let's talk about the alternative, this nanopore electrometry. How does it actually get around these problems?
Well, nanopore electrometry flips the script entirely on what you measure. Instead of measuring that bulk current flow across the whole membrane, the total flow through the pipe,
Right.
it focuses on measuring the modulation of the local electric field right near the pore caused by the molecule as it passes through.
Okay, hang on. So instead of measuring the overall drop in water level when something goes down the drain, you're putting a tiny, super sensitive probe right next to the drain, watching the electrical ripples the object creates as it goes past.
That's a great analogy. Yes, it's a very localized measurement. And the key insight is that the local electric field is inherently sensitive to the specific charge distribution of the molecule passing by, not just its overall size.
Ah, so it picks up on those positive and negative patches you mentioned.
Potentially, yes. Which means the signals you get from electrometry could contain significantly more information, a much higher information capacity compared to just watching the overall current dip.
That sounds promising. Did the researchers just theorize this, or did they model it?
Oh, they modeled it extensively. The paper uses really comprehensive computational models. Things like multi-physics continuum models solving complex equations like Poisson-Nernst-Planck that describe ion flow and electric fields.
Okay, standard but powerful physics modeling.
Right. And they combine that with all atom molecular dynamics, MD simulations. So simulating, for example, single amino acids moving through a tiny graphene nanopore in a salt solution, and tracking the electric field changes atom by atom.
So the physics and the detailed molecular interactions, they both suggest this should work in principle. But how do you actually build a sensor that can measure an electric field on that tiny scale right next to the pore? That sounds technologically demanding.
It absolutely is, and the paper acknowledges this. The experimental realization really depends on having these highly localized nanoscale electric field sensors.
And do those exist?
Well, progress is being made. The researchers specifically point to advances in sensors based on something called nitrogen vacancy, NV centers in nanodiamonds.
Nitrogen vacancy centers in nanodiamonds. Okay, that's quite a term. What are they?
So think of a tiny, tiny diamond crystal, a nanodiamond. An NV center is a specific defect inside that crystal lattice. It's where one carbon atom is missing and replaced by a nitrogen atom right next to an empty spot, a vacancy.
Okay, a specific flaw in the diamond structure.
Exactly. And this particular flaw has remarkable quantum properties. It acts like an incredibly sensitive atomic scale sensor. It can detect minute changes in local electric and magnetic fields, even at room temperature.
Wow. So you could potentially place one of these NV center nanodiamonds right near the nanopore opening.
That's the idea. They are the kind of critical enabling technology you'd need to actually build and test nanopore electrometry in a real experiment.
This shift to local field sensing is clearly powerful. But you mentioned something earlier about charge polarity, that this new method can tell positive from negative charges in a way the old one couldn't. That sounds really significant.
Yes, this is perhaps one of the most unique aspects. It comes down to the inherent asymmetry of the electric field measurement. Remember we said the ionic current blockade is generally symmetric.
Right. A blockage is a blockage, whether caused by something positive or negative of similar size.
Exactly. If all you measure is the total current reduction, a positively charged molecule and a negatively charged one might look very similar if they block the same volume. It's like Only seeing the shadow, you know something's there, but not its charge.
Okay.
But the electric field sensing described in the paper is fundamentally asymmetric. The simulations showed something fascinating. If you put your NV sensor, say, on the top edge of the pore,
Yeah.
it might turn out to be highly sensitive to positive particles, vacations, giving you a big, clear signal change when one goes through. But for negative particles, anions, the signal contrast might be really small, almost flat, regardless of how strong that negative charge is.
Whoa. So the sensor location itself makes it selective for one type of charge.
Yeah.
That's amazing control.
It's not just location, it's also the applied voltage bias. But yes, the key is this asymmetry, and you can apparently reverse and tune it simply by changing where you put the sensor, maybe move it from the top edge to the bottom edge, or by flipping the direction of the external voltage you're applying across the pore. You could potentially tune it to be sensitive only to cations or only to anions.
That ability to selectively pick out positive or negative charges on the fly, that's completely different from just measuring the total blockage.
Completely unprecedented with standard ionic current methods.
And the detailed simulations, the molecular dynamics ones, they confirmed this selectivity actually works at the atomic level.
No.
With all the messy reality of ions and water jostling around.
They did. They simulated 3 different amino acids going through the pore. One positively charged, lysine, LYS, one negatively charged, glutamate, GLU, and one neutral, tyrosine, TYR.
Okay, a good test set.
And the results were clear. The charged amino acids, LYS and GLU, produced strong, distinct modulations in the local electric field signal, and importantly, the signals looked different depending on the polarity, positive or negative.
And the neutral one?
The neutral tyrosine. It barely caused a ripple. It was hardly distinguishable from the background noise in the electric field measurement.
That really drives it home. It's not just measuring size or volume. It's genuinely sensitive to the charge itself.
Exactly. High selectivity for charge.
Okay, so we have potentially higher information capacity, charge selectivity. What other practical advantages come from being able to measure locally like this, right at the pore itself?
Well, thinking about the setup, the localized measurement brings a couple of really big structural and environmental benefits. First is the possibility of self-referenced error correction.
Self-referenced. What does that mean in practice?
It means you don't have to rely on just one sensor. You could place multiple local electric field sensors around a single nanopore, maybe one at the top edge, one at the bottom edge, perhaps even one near the middle, inside the pore wall if technology allows.
Okay, multiple viewpoints on the same event.
Precisely. Each sensor location will give you a slightly different signal profile, a different view, for the exact same molecule translocating through the pore at the exact same time.
Ah, I see where this is going. You can compare the signals.
Right, you can correlate these simultaneous measurements. If one sensor gives a weird reading, but the others look normal, you can flag that as potential noise or an error. It allows for internal signal calibration in real time.
And could it help with that speed variation problem? Molecules speeding up or slowing down as they go through.
That's a key potential benefit. Those fluctuations in translocation velocity are a major headache for high-resolution ionic current measurements. But if you have multiple sensors tracking the same molecule, you might be able to use the timing differences between the sensors to actually measure and compensate for those velocity changes.
That would dramatically clean up the data.
Yeah.
Okay, so error correction, velocity compensation, it's huge for data quality.
Sure.
What was the second big advantage you mentioned? The environmental one.
Yes, this one. This is potentially the real game changer for where you can use this technology. It's the possibility of electrolyte-free operation.
No electrolyte. No saltwater solution.
In principle, yes. Nanopore electrometry measures the molecule's effect on the local electric field. It doesn't fundamentally rely on measuring the flow of ions from a bulk electrolyte solution through the pore in the same way ionic current does.
But don't nanopore sensors always need to be in some kind of conductive liquid? That seems like a core requirement.
It has been for ionic current methods. That need for a stable, usually high-salt liquid environment has basically tethered these devices to, well, laboratory benchtops or carefully controlled settings.
Right. You need pumps, fluidic, stable temperatures.
Exactly. But if you can remove that hard requirement for the liquid electrolyte, suddenly the possibilities explode. You could potentially take the sensor almost anywhere.
Anywhere. Like what?
Well, the paper explicitly mentions entirely new frontiers. Think about direct monitoring of airborne targets.
Like viruses or pollutants in the air. Just sampling air directly through the pore.
That's the kind of application it could open up. Or using these sensors in extreme environments where maintaining stable, complex liquid handling systems is incredibly difficult or costly.
You mean like space stations,
Yeah.
remote polar research sites,
Ah,
deep-sea exploration.
precisely. Places where rugged, simple, low-maintenance sensors are needed. If you can get rid of the need for the liquid lab part of the system, molecular detection becomes potentially much more robust and deployable in the field.
Okay, so just to wrap this up then, this deep dive into nanopore electrometry. It's really painting a picture not just of an improvement, but a fundamental shift. We're moving away from measuring the bulk flow, the total current drop,
Right, the fire hose measurement.
and moving towards these highly localized, information-rich measurements of the electric field right at the point of action. It seems to offer a path to faster sensing, much more detailed signals, and remarkably, far more versatile applications.
I think that's a fair summary. And the implication is profound. The bottleneck in the old technology was ultimately the physics of the molecule's transport speed versus the measurement rate.
Yeah, the million bases per second problem.
Exactly. But now, with electrometry, the practical performance limits seem likely to shift. They'll be set instead by how good we can get at making and reading those nanoscale electric field sensors, the spatial resolution, the temporal resolution of things like those NV centers.
So the challenge moves from controlling the molecule's speed to engineering ever better detectors.
That seems to be the new frontier. The focus shifts from the transport physics to the sensor physics in engineering.
That gives the field a very clear target for the next decade or so. Which leads to a fascinating thought for you, the listener. If detecting individual viruses, proteins, DNA fragments, if that no longer requires maintaining these complex, wet laboratory setups, how quickly could these kinds of advanced, rugged sensing devices move out of specialized research labs? Could we see them appearing in, say, environmental monitoring, industrial process control, or even consumer diagnostic tools much sooner than we thought? That's definitely something to watch.
Absolutely. The potential applications become much broader.
We'll leave you with that thought. Thanks for joining us for this deep dive. We'll talk to you next time.